Thank you very much indeed, Mr. Chairman, for your wonderful introduction. And this hoping that my lecture will be as enticing as the introduction. Thank you very much indeed to the organizers for inviting me to this wonderful city again, for the wonderful sunshine, food, and hospitality. Always a pleasure to do pharmacology in civilized environments. Thank you very much for that. I was given a difficult task to really talk about what are the problems that we're facing in psychopharmacology at the present time, particularly in terms of new drug developments, and why possibly do we have those problems. So what I want to do is, over the next half an hour, 40 minutes, is to give you some of my ideas of why we have problems, and they may well differ from some of your ideas, but the whole purpose of this talk, I think, is to stimulate discussion, and that is what good psychopharmacology research should be about. So, I think we're all aware, wherever we are in the world, that there is a major crisis in getting new drugs for treating mental ill health. And it's relatively new. Looking around this audience, I can see that most of you have been practicing psychiatry for many years. And you know 10 years ago that in, for example, the field of antidepressant research, we had passed on from the old-fashioned monoamine oxidase inhibitors and tricyclic antidepressants and nyanserine, and we were coming into the era of the serotonin drugs, in particular the SSRI antidepressants, the SNRI antidepressants, and we thought that this was going to really herald a new era, particularly in the treatment of depression, anxiety disorders, and compulsions, and so forth. And to some extent, it did. But now we look at the situation today, and we think, well, where are we going? These drugs, although they were very useful and an improvement, are by no means as good as we would hope they would be. And what I've done is to put onto this slide two areas. First of all, if you look at all of the major drug companies, the number of new drugs in all areas of psychiatry and overlapping neurology are not very good in terms of new entities. And if one actually looks at some of the figures that I've managed to get, international figures from major drug companies, you can see here that in the major areas that we're interested in and still require good medication for treating effectively depression, dementia, schizophrenia, anxiety disorders, addiction, and eating disorders. These are the total number of entities which are coming through to stage 3 trials. That does not mean they will all be launched and marketed as drugs for treating these particular conditions. We have a long way to go. Now, what are the possible reasons? Again, there are many reasons. I don't profess at all to have all the answers, certainly not. But if you actually look at the list, it's fairly obvious. Economic factors play a very important part. And what has happened in all of the multinational drug companies is there's been a contraction of the industry. There are fewer and fewer large conglomerate companies. If you take a company like Pfizer, for example, then as you know, Pfizer has taken over big companies in recent years like Upjohn, Wyeth, and so forth. And these now are all, of course, under the same brand heading, with the same basic research personnel and therefore research interests. So this is bound to have some sort of impact when you're rationalizing research in a large company on the type of entities and areas which you're going to work in. So it means, therefore, that there's a concentration of research effort into specific regions and away from other regions where there was originally quite a lot of competition to see which would get behind and between some of the large firms. I think there are scientific reasons, and this concerns many of us, because this is the area in which we are working every day. One has to look at the whole history of psychopharmacology. The real revolution in psychiatry, in my opinion, took place between the 1950s and the 1960s. And the major breakthrough, the major drugs that we had in those days were discovered by a process of serendipity, which is just a fancy way of saying pure chance and good critical observation based on that chance. And in fact, all of the subsequent developments in the major areas of psychopharmacology were based on refining the profiles, the biological profiles of those original drugs. I'm talking, of course, about the monoamine oxidase inhibitors, and particularly the tricyclic antidepressants like imipramine, the antipsychotics such as chlorpromazine, the anxiolytics such as diazepam and chlordiazepoxide. And if you think about it, all the subsequent developments were based essentially on the pharmacological properties of those substances. What has been very limited and still is very limited is our understanding the psychopathology of all of the major psychiatric disorders. Whereas in other areas of pharmacology, such as cardiology, we can talk quite meaningfully about drug targeting. A heart, whether it's in a rat, a dog, or a human, basically functions as a sophisticated pump. That is not the human brain. And here we have, I think, a major problem, because until we can start, I think, delineating the underlying psychopathology, it's extremely difficult to rationally treat that as a target and develop specific chemicals which are going to somehow normalize some of the outer functions. The second thing, what do we do? We can't do experiments on humans, certainly not on the human brain. Oh, we use animals. And of course, it really is quite ridiculous, and I'm speaking from experience, having spent 40 years in the laboratory, working largely with animal models, that here you have a rat with a brain weight of about 1.3 grams, and you're comparing the complexity of the human brain of something like 1.4 kilograms with an animal with a brain of 1.4 grams. It doesn't make sense. So is it surprising, therefore, that no matter how sophisticated our scientific studies on rats and rat brain and rat behavior, that we often get it wrong? So therefore we do more sophisticated things. We now go into the area of psychogenetics, molecular neurobiology, and unfortunately, we often get that. When we're talking about genetics, which has taken billions of dollars of research funding in most countries, certainly in Western Europe and North America, and then you start looking here at the proteomics of schizophrenia and depression, and this is a paper that was published a couple of years ago now, and you can see here that all of those proteins, which represent different very important biological processes in the brain, the enormous overlap between, on the one hand, controls and schizophrenia, but most of the changes lie outside. So we really do have to go in trying to extrapolate from our genetic basis into, I think, a meaningful basis of trying to understand psychopathology. It shouldn't stop us trying, it shouldn't stop us trying to get better at it, but to believe that this is going to be the answer in the foreseeable future is, I think, very optimistic. But I'm old, maybe that's just my view. Okay, so let's go back to the beginning. We still have a very, very simple view of drugs for treating psychiatric disorders. In the old days when I started, which would be way back in the 1950s, the brain chemically was very simple. It consists of monoamine neurotransmitters, noradrenaline, dopamine, and serotonin. We could explain everything in psychiatry in terms of those three amines. The few drugs that we had at that time, which seemed to affect the brain in some way, we could explain them by how they affected noradrenaline, serotonin, and dopamine. And so therefore we came up with crazy ideas, like depression was caused by a deficit in serotonin and noradrenaline. Schizophrenia was caused by too much dopamine. Anxiety was caused by too much noradrenaline. In those days, we didn't know much about GABA. So therefore, we could understand what the drugs were doing. Having just been discovered then, we had imipramine, we had chlorpromazine, and later we had diazepam. But of course, there aren't just three neurotransmitters in the human brain. There's well over 54 different neurotransmitters and neuromodulators. They all interact in a very complex network of changes. Why, therefore, do we have all of those neurotransmitters if only three really matter? Perhaps we've got it all wrong. And this just gives you some idea of how it used to be. Beautiful, right in the center there, we have dopamine, noradrenaline, serotonin. In psychiatry, we've got our motivation and pleasure associated with dopamine. We have energy, alertness associated with noradrenaline, and we have obsession, compulsions, and to some extent mood associated with serotonin. All interlocking. Wonderful, wonderful. Just take two other neurotransmitters, glutamate and GABA, two amino acid neurotransmitters, which modulate at least 50% of all synapses in the human brain, as distinct from approximately 5% of synapses in parts of the brain modulated by the amine transmitters. And what you'll see there is none of those amines function without some input and modulation by an excitatory and an amino acid. And that's just 5 of anything up to 54 different modulatory substances working in your brain at the present time. And so what causes depression? I wish we knew. And it's very interesting if you look from the 1950s through to the present time, and you look at the major drug discoveries, in this case depression, during those periods. We explain the pathology in terms of interpreting what the drugs do to treat the symptoms. So, imipramine and monoamine oxidase inhibitors, once we knew roughly how they worked in pieces of rat brain, then we said that was the basis of the monoamine hypothesis. And then we realized that it's not as simple as that, because if that was true, these drugs would work straight away. They don't. They take weeks and months to work. So therefore, something else must be happening in the brain. And we then started to look at and understand more about receptors upon which the information is passed from one neuron to another. And having done that, we realized it's much more complex than that, because the receptors have to speak to the nucleus and have to change the expression, protein synthesis, and so forth. That requires signaling mechanisms within the actual neuron itself, so the information has to filter through. And then, of course, so therefore that could be another problem in terms of psychiatry and psychopathology. And then, of course, it becomes even more complex, because the brain is not just a number of wires connecting different bits together. We have tracks which interact and talk to one another and cross-talk and modulate one another. We have a neuroplasticity. We have an underlying circadian rhythmicity, which makes it even more complex to understand. And so you can see, by and large, how we explain or hypothesize about the nature of depression is really the mirror image of the drugs and treatments that we're using, in the hope that I will tell us something about the pathology. Okay, so really I think I'm just trying to create a picture of why working in this area as a pharmacologist and trying to create new entities is much more difficult than any other area of clinical pharmacology. The brain is a difficult and complex and challenging organ. And so let's look a little bit outside the box, and that's what I want to do in the next minute or two. Let's move away from the aimings and see where that's going to lead us. Maybe we're going to just as big a mess, but at least we know where we are at present, or we've not gone far enough. And what's coming through in all of the sort of neuroscience research at present are three words: neurogenesis, neuroplasticity, and neurodegeneration. And these, of course, interact, but it tells you something about the plasticity of the nervous system and how, in fact, environmental challenges and so forth can change, improve, or disimprove that plasticity, and thereby the mental health of the individual. And there's another thing we have to think about: the so-called hygiene hypothesis of mental disorder. I'm not going to get into this. It's a very, very interesting, relatively new area, a new concept. But basically, if we look at the immune system and the way the immune system plays a very, very important part, not only in preserving the integrity, preventing infections and so forth, but also profoundly affecting brain function. And so we now have a very new area of psychoneuroimmunology, which is coming into increasing prominence and indeed is part and parcel of some of the symposia here in this Congress, I'm very pleased to say. And so consequently, the way we interact with our environment, the way our body is protected against infections and so forth, the way our immune system is either activated or not, has quite a profound effect upon ultimately how our brain works, and I'll touch on this again later. So what causes depression? Just to sort of start the thinking going, The answer is we don't know. There are a number of possibilities. A primary deficit in monoamines, yes, undoubtedly something happens to our amines. Deficit in other transmitters, like excitatory amino acids. Yes, indeed, that may happen. But what about repair mechanisms, the so-called neurotrophins, brain-derived neurotrophic factor? There's a lot of interest now in these little peptides, which are really very, very important in maintaining the integrity of neurons and repairing damaged neurons, which invariably occur during normal life. And if obviously there's a fundamental problem in the repair mechanisms and affecting these peptides, this can have a profound effect upon the integrity of the nerve networks. And then we have a relatively new area, which is, again, I think, of growing interest, and that is the role of these accessory cells around the neurons, the so-called neuroglia and the dendroglia. And in the old days, we used to think of them as sort of packing cells. They somehow stop the neuron from banging about in the brain and getting damaged. Much more important than that. And certainly if one looks at the astrocytes are extremely important in basically controlling the metabolic activity of neurons. So the actual movement of glucose across the neuronal surface, to a large extent, is dependent upon the normal functioning of the astrocytes. So these secondary cells, in fact, to some extent, even start to determine whether or not a neuron can function in a healthy way and normally. And then we have the microglia, which are also part of the neuroglia network, which, of course, can produce inflammatory mediators. And these inflammatory mediators, the inflammatory cytokines, can have a very damaging effect if present in excess within the brain. And there is now very clear evidence that in chronic major depression, indeed, that is precisely what happens. Associated with both major depression and chronic schizophrenia, there is indeed a chronic low-grade inflammatory change within the brain. Often, not always, but often related to overactive microglia. So this will have an impact on the integrity, again, of individual neurons and neural networks. And then we have, of course, the consequences of that. The presence in the brain of neurotoxins, which not only are in products of normal metabolism, of purine metabolism, of amino acid metabolism in the brain, but more importantly, the end product of the activation of some of the biochemical pathways caused by pro-inflammatory cytokines. And there, in fact, is evidence, which I don't have time to go into, but last year this turned up in some of the symposia that we were involved in here, particularly some of the work of my colleagues in Munich and Jens, showing very clearly that the metabolism of tryptophan down the kynurenine pathway produces a highly toxic end product, quinolinic acid, which activates the glutamatergic system and is ultimately responsible for the death of neurons. So here we have in the brain an endogenous neurotoxin, which is also part of the pathology, the chemical pathology. So there are many possibilities. All may be right, all may be wrong, and there may be others. But these are possibilities which, if we're going to talk about breaking through the barrier to produce new drugs and so forth, have to take these things changes into account. So without going on any further, I think that we, in terms of the latest and the thinking in terms of neuroscience, we have to think in a different way about the brain, about its pathology, about not only how the drugs we have work or don't work, but how new drugs could possibly come in to help us overcome some of these problems. So, just looking at this one or two. And the other aspect which clinicians are often aware of, particularly in their middle-aged depressed patients, is that even though their mood state improves, even though they can get back to work and fairly normal life, the cognitive deficits remain. Is there something still that even good antidepressant treatment does not, in fact, reverse and improve cognitive deficits? And if so, is that part of the underlying pathology that our drugs are not touching? And furthermore, the other big problem is many of our patients, our middle-aged elderly patients, who had a lifetime of depression, very quickly deteriorate into dementia, particularly Alzheimer's disease. There's a much, much higher probability of this happening. And so therefore it raises the question, are antidepressants limited as neuroprotective agents as well possible as antidepressants? And we have a lot of them. I'm not going to bore you, you know all these. Starting with the old-fashioned monoaminoxidase inhibitors, tricyclic antidepressants, a whole number of SSRIs, SNRIs, receptor antagonists like mirtazapine, noradrenaline reuptake inhibitors like reboxetine, which is not marketed, which is a great pity. Bupropion, dopaminergic type drug, the latest, agonality, which is manufactured as a melatonergic agonist. In fact, it is a 5-HT2C antagonist, which accounts for its therapeutic action. The effect on melatonin certainly helps in terms of the circadian rhythmicity and possibly sleep, but it's certainly not due, or doesn't play a major part in this antidepressant action. Otherwise, melatonin, which would buy a health food shop, should be a reasonable antidepressant, and it is not. So, what's the problem? A number of problems. We really should look at schizophrenia, depression, not in the singular, but in the plural. We're talking about depressions. We should be talking about schizophrenias. These are very complex diseases. They're very heterogeneous diseases, but both clinically and certainly biologically, we say they're all the same. In all probability, they're not and could be very different. Success? Well, yes, because the SSRI antidepressants have been safe and reasonably successful. There's very much the view until recently, one drug suits all. You've got a bit of anxiety, you're a bit depressed, fine. You're a little bit OCD, okay, take an SSRI. Cures everything, come back and see me another time. It's not like that. And then, of course, we've got the rebound. The media, and I can't talk about Turkey, but the media, certainly in Western Europe and the UK, to a far lesser extent, fortunately in Ireland, but certainly in the UK, there's a very, very strong anti-psychiatry media. Anything which goes wrong in psychiatry, they love it. It appears on television, in the newspapers, all sorts of clowns are interviewed to tell the general population how dangerous these drugs are. There's a guy who has become extremely famous, a clinical psychologist called Erwin Kirsch. Only the most severely depressed patients, in fact, ever respond effectively, possibly to an SSRI. These are dangerous drugs that do not work and can even precipitate suicide. story which the media loves to carry. Unfortunately, his statistics are not very good, and the Journal of Psychopharmacology and indeed the World Federation of Biological Psychiatry Journal has published statistical analyses showing to use biased statistics to come to his erroneous conclusion. But in the meantime, the damage is done, and you cannot expect television channels and popular newspapers to come back and say, well, it was a case of statistics. Damage is done. So what's new? Not a lot. In recent years, we've had escitalopram, which certainly is important in at least pharmacologically in pinpointing the importance of the so-called enantiomers, the aspects of a molecule which lead to its pharmacologically active components. And then we have metabolites. We have venlafaxine. But then if you look at this metabolite, there's venlafaxine, then you have another drug which is more potent on the noradrenergic transporter than the serotonin transporter. So therefore you market that as a new drug. And then you've got reformulations of old drugs. So in Eastern Europe, trazodone is coming back into prominence again, even though it was a very old drug. We were working on this drug in the 1980s. And then, of course, you've got augmentation strategies, which are very important, of course, for treating patients who show therapy resistance. But again, lithium's been around for a long time, and now we have, of course, the safer atypical antipsychotics. Whether they're better is another matter. And then we've got novel drugs. I've already mentioned agomelatine, which, as I say, is really a 5-HT2C receptor antagonist, which, by the way it works in the prefrontal cortex in particular, increases the efflux of noradrenaline and dopamine. So although it acts on a serotonin receptor, it is not a serotonergic drug. In fact, it's a noradrenergic dopaminergic drug, amongst other things. Vilazodone, again, this is coming from the Leonard stable, where it's a partial agonist of serotonin 1A receptors, so therefore it's sharing properties with very old-fashioned drugs like buspirone and bisaparin, together with an SSRI antidepressant. But what they're all doing now is saying, well, specificity is a thing of the past. Now we have to go for blockbuster drugs which do everything. So at least go and hit all of the aims. And there are none of these drugs coming along which do precisely that. And this just gives you some idea, and this is just one company, this is a Unmet company that started work with citalopram, and then found that the enantiomers were not equally active, that it was the S-enantiomer which was the active one, and indeed that was then launched as a very selective SSRI, which is still with us and a very good drug. We then have vilazodone, which I've already mentioned, saying, well, what we want then is to bring in some of this more anti-anxiety effect on the cell body. Now we have an even more complex one, vortioxetine, which is due to be launched, I think, in some countries fairly soon. This is a SSRI. It also blocks noradrenaline reuptake inhibitor. It then hits a whole number of receptors that blocks them. It also increases acetylcholine and histamine in the brain. It's a very interesting molecule. I mean, it's doing all sorts of things. And so what's the idea there? The idea there, we have an antidepressant. But also we possibly have a drug that does nice things with cognitive dysfunction. So maybe somebody in the early stages of Alzheimer's, this could help them. Now we have an even more complex one, which is again just being developed, 10-deoxetine, which, as you can see, does everything that you could wish for in terms of the monoamines. So what are pharmacologists doing? They're still basically peddling the old non-aiming theory, but in a more sophisticated way. Maybe it helps. We don't know. So, can we get outside that box? This, I think, is the most exciting thing that's happened for a long time, looking at the glutamate system. Well, it's interesting because going back again to the way in which our initial drugs were discovered in psychiatry, the same thing's happening to some extent here. Amyotrophic lateral sclerosis is not the sort of disease which most psychiatrists come along every day, across every day in their clinic. Very nasty disease, but patients who are treated with riluzole, which does have some benefit symptomatically, will notice that riluzole has an antidepressant effect. And riluzole is, in fact, quite a complex antagonist of some of the ionotropic glutamate receptors. And now there are a number of other drugs, again, discovered accidentally as side effects in neurology, which do roughly the same thing. So that's one approach. Let's look at the glutamate system. The other approach is to look at the endocrine and immune system and see if by modulating the endocrine and immune axis, one can, in fact, also start to improve mood states. Because it's been known for a very long time that stress, of course, has a very big impact upon mood. And so here we have a whole number of compounds which look at, which modulate glucocorticoid receptors, vasopressin receptors, also modulate the production of some of the inflammatory mediators like prostaglandins and so forth. And these indeed might have some, and do indeed have some, antidepressant type activity. So, if we look at why all these antidepressants, no matter what they are and what they seem to have in their pharmacology, the ones we've had so far, why do they all roughly work to the same extent and not one being particularly superior to the other? Well, basically, they all work on the same basic subcellular mechanisms. They eventually all affect the so-called second messenger Krebs system and therefore modulate nuclear expression. Now, what about other possible mechanisms? And particularly when we're thinking in terms of more rapid types of targets. And this just gives you a very crude diagram of the mechanisms going from the receptor on the outside of the neuronal membrane, coming through to gene expression, leading to neuroplasticity and survival and so forth, which account for most of the current antidepressants and the way, and as a summary, how they basically work. What I think interests many of us, and I know a lot of people here are interested, was this particular report that came out now in 2006 originally, which both excited and shocked us. Randomized, placebo-controlled, double crossover study of ketamine in the treatment of resistant depression. Those of you who read this paper will know, and I can imagine, the impact it made on us at that particular time. First of all, these were highly resistant patients who had immediately responded to electroconvulsive shock therapy. And when they were given an intravenous injection of ketamine at a dose which was about 1/4 of the normal anesthetic dose, and therefore didn't produce obvious signs of disorientation and psychotic-like states, produced within less than 24 hours a rapid improvement of mood which was sustained. Now these were very, very resistant patients that were not responding to anything. And suddenly, not only did they get their mood state improved, but it was sustained often for a week, sometimes longer after a single injection. Now when we read this and heard this, none of us believed that this was just a miracle. This doesn't happen in clinical neuroscience. But it was repeated, and double-blind conditions and all these sort of things, all showing roughly the same type of change. Some responded very rapidly, others more slowly, some showing a sustained response for a longer period than others and so forth. But by and large, it was just a miraculous finding. So then the question is, what is going on here? This is a drug, a complex drug, which affects both the N-methyl-D-aspartate and the NMDA glutamate receptors in a way which no other antidepressants have been shown to do. And of course, as I say, it was repeated and so forth. And then eventually, we came to the conclusion that this drug was very peculiar because it was working in a completely different mechanism from the mechanism which seemed to underlie the way in which conventional antidepressants work. And it could, in fact, without going into any of the biochemical details, it could, in fact, be due to the fact that it does switch on the so-called mTOR system, which leads to increased synaptic activity, increased spine density, and therefore improves rapidly the integrity and functioning of the synaptic membrane. And this was a very, very unique type of biological change. Now, since that time, of course, why don't we use ketamine? Well, ketamine has been used and is being used in a hospital setting. It's a very nasty drug. It has side effects. It can only be given intravenously for full efficacy. And, of course, it's not without risks. So we need, obviously, to use this information in drug development. And there are now, in fact, one or two drug companies which are producing compounds which seem to particularly target that mTOR system. And at least some of the early clinical data suggests that indeed this might lead to a new generation of antidepressants. So that's one big thinking outside the box that could be a very important development for the future. Not based on drug company research. This was the National Institute of Mental Health Basic Research Group, which is in fact doing this. And thinking outside the box and now trying to get new ideas as to how we can produce new drugs. And there are other crazy ideas coming through. Is there such a thing as a leaky blood-brain barrier in depression, whereby some of these pro-inflammatory cytokines not only are produced in the brain by stress, but also get into the brain and cause further damage? And if so, what are the consequences? And there are changes we now know biologically in patients with depression involving the hypothalamic pituitary adrenal axis and enhancing the stress response, with inducing the production of the neurotoxin, quinolinic acid I mentioned, activating the glutamate pathways to actually cause a destruction of neurons, and of course having an effect on numerous neurotransmitters, including the monoamines. So there are a whole number of possibilities here that maybe by modulating chronic low-grade inflammation could be, in the long term, another way of modulating these effects. So there's possibilities here of looking differently at the brain and producing new types of drugs. This is a list of some of them. And the interesting thing is anti-inflammatory drugs. We need to get better ones that penetrate the blood-brain already, but already in terms of proof of concept, there are a number of very good double-blind clinical trials shown that a drug like celecoxib used in the treatment of arthritis does in fact, as an adjunctive treatment with antidepressants that are not therapeutically as effective as they should be, does improve the efficacy of those drugs. So there's a whole number of possibilities there. I'm not going to deal with other areas in so much detail because I'm more pessimistic. I'm much more pessimistic if one looks at, for example, the antipsychotic stuff. The Senaprin, what's the Senaprin? Senaprin, when I was working in the industry in Organon way back in the early 1970s, we were working on these tetracyclic compounds, including this drug, which eventually became the Senaprin. And we were developing it as an antidepressant at that time, I seem to remember. And structurally very similar to mirtazapine. It's a very complex drug, serotonergic drug, a weak noradrenergic drug, and so forth, and so forth. Is it an antipsychotic? Yeah, it has antipsychotic activity. Is it any better than the ones we have? Probably not, but it's there. Iloprone is a risperidone analog. Is it any better? Probably not. Not much evidence that it is. There are one or two new ideas coming along. Lilly, I think they've stopped development at present. It is temporary, but they were developing an interesting glutamate metallotropic receptor antagonist, which seemed to do wonderful things in Russia. But when the drug was moved over to America for clinical trials, it didn't work at all. Now, was that due to the Americans being different from the Russians, which I'm told they are? Or was it that they didn't like it, or what was going on? We don't know. But clearly, it seems very extreme anyway. Works in one country but not the other. So maybe that will come back. But there is sort of some interesting movements going on there, again, with the glutamate system. And then anxiolytics, again, it's very boring. There's not much going on. It's new. You know, we've got gabapentin, we've got pregabalin. They were being developed for the treatment of pain. They weren't being developed at all for the treatment of anxiety. Just so happens that it was found to have some sort of anti-anxiety action. And so now we have a very, very expensive anti-anxiety drugs with side effects, of course, which seem to work differently, and maybe it is going to open up a new area. Again, they affect too much. Dementia is the real, I think, challenge, and you all know this. Dementia for years and years and years. If you've got plaques and tangles and they build up, then you become demented. Anyone over the age of about 70, including myself, has plaques and tangles. How many of us demented? I don't know, maybe I'm demented. But still, we function. And the point is that this is, I think, we can now produce wonderful models. We can express the human beta amyloid gene in the mouse, and we can produce mice with human beta amyloid. And you think that's the best model. Not perhaps one, but it would be so far. Because the mouse is not human. So beautiful stuff. And we know the pathways. We can block the pathways. We can produce specific drugs that do that. We can passively immunize human beings against beta amyloid. Unfortunately, of course, one can always argue that the brain was too far damaged, but you can get rid of a lot of those plaques. It doesn't do anything to the normalization of brain function. And so there is a possibility that this whole amyloid story was reflecting a consequence rather than an actual cause of Alzheimer's disease. And we wasted billions and billions and billions of dollars chasing the wrong hair, as we say. there are two novel approaches. Dynambon was a Russian discovery again, which did all sorts of interesting things. I remember attending lectures by Russians on this. It worked on histamine receptors as an antagonist. It seemed to do very nice things to oxidative stress, and reduces oxidative stress, and indeed some of the initial clinical studies were very promising and very exciting. They couldn't be replicated, certainly not outside of Russia. So therefore we're going back to the old story of looking at acetylcholinestrases in a more sophisticated way, with butyrylcholinesterase inhibitors combined with monoamine oxidase B inhibitors, and therefore try and improve the cognitive enhancing properties and so forth. These concepts are not new. They might be better than what we have. They might slow down the process of deterioration. They're certainly not likely any more than what we have with donepezil to stop it. So we still have quite a way to go here. And the other possibility, if information is the important part, why don't we all start taking aspirin when we become teenagers, particularly if Granny had Alzheimer's disease? Unfortunately, we have to do it for a very, very, very long time, by which time we might have other problems with the complexities of the non-steroids that we currently have. But certainly we do know that giving non-steroid anti-inflammatories to patients with Alzheimer's does nothing. And then we've got funny drugs like modafinil, which is an amphetamine-like in terms of structure, but this isn't amphetamine. And it certainly doesn't seem to have an abuse potential. And it's a very interesting sort of alerting drug for treating daytime sleepiness, particularly associated with narcolepsy. Any of you traveling from one side of the world to the other, I'm not talking about jet lag, but if you just get, as I do, sometimes a bit tired when you get to the other end, you take one pill of modafinil and that certainly puts you right so you can function for the day. Very interesting drug. We have no idea how it works. It's extremely complex. It seems to affect amine, GABA, histamine, hypocretin, which probably affects sleep-wake cycles and so forth. So here we have another case of a drug which we're using, which is effective, appears to be safe. How it works, we still really don't know. And then the big flurry in Europe now is what should we do with cannabis? Should we legalize it, as everyone seems to be smoking it, or should we treat it still as a drug which should be very strictly controlled? I'm not interested in the legalization. What I am interested, however, is in the pharmacology of this very, very fascinating group of molecules, the so-called endocannabinoids, the naturally occurring endogenous cannabis derivatives in our brains and what those drugs are doing. And here we have natural ligands for them. We know a lot about them. We know a lot about their physiology too. And they're very, very, very complex molecules. And so it certainly wouldn't do to just go in and say automatically, let's launch drugs that seem to modulate them. We need to understand the physiology much better. And we know that there are profound changes in these endogenous substances, in inflammation, in pain, in neurological disorders, including dementia disorders, and in psychiatric disorders such as depression and anxiety. So there is a possibility here that we should be looking at the cannabinoid system for new drugs of the future. And we can do a lot of things in mice and so forth. All of you have heard of the drug developed for smoking cessation, ribonomab. which is a cannabinoid receptor 1 agonist for smoking cessation, it had to be withdrawn because it actually precipitated depression. But there are others coming along who are treated with obesity. So maybe this is, again, an area where we should be looking at in terms of psychiatry. And then what's on the horizon is a whole number of non-drug approaches, non-psychotropic drugs in the future. Stem cells might be useful to Parkinson's. Is it going to be useful to Alzheimer's and related psychiatric disorders? I doubt it. The gut-brain axis and the future of nutri-pharmaceuticals. This is a very hot area now. Looking at the way the gut bacteria or products of gut bacteria affect profoundly brain function. And there's in fact some very interesting experimental work about to be published showing that if you look at the gut bacteria in animals and possibly humans also that develop a metabolic syndrome in response to atypical antipsychotics. If you change the gut bacteria by sterilizing the gut and then giving more neutral type bacteria, you lose the metabolic syndrome in those patients. And it does appear that what the drug is doing in those individuals is changing the composition of the gut bacteria, which then have secondary effects on the metabolism of those individuals. Again, a whole new level of complexity in the sort of way we treat patients. Medicine, again, already coming into cancer research, and optogenetics and gene therapy. All of these are techniques which are possible within your lifetime, if not mine. So what can we expect? All this wonderful stuff may not. We can expect fewer psychotropic drugs and even fewer non-drugs in the immediate future. We're going to see a greater emphasis by regulatory agencies on generics, cheap psychotropic drugs. We're going to see the increased importance of small biotech companies and university-based research institutes in developing new ideas and novel targets. The problem is that has to be carried forward into industrial production. That's where the block will be, because it's risky. And I think we also have to acknowledge that serendipity is the key factor so far in psychotropic drug development, because we don't understand the basic psychopathology of the complex diseases that we're treating every day as psychiatrists. And until we begin to understand that in a more rational way, it's going to be increasingly difficult to do anything about it in terms of really novel drug development. And so I leave you with one slide. Our biannual congress, the 29th Legion Internationale Neuropsychopharmacology Congress, is being held in Vancouver in June, and the beginning of June. You're all very welcome. Sorry I can't give you any support. I've had difficulty in getting support for myself, but I assure you, if you get there, it's 3.5 days of really exciting reviews, updates, poster sessions, and so forth. And as you all know, Vancouver is a very civilized and very pleasant place. So thank you once again, ladies and gentlemen, for your attention. Thank you for having me. I hope I don't bore you.